So hopefully you were all able to grab a cup of coffee and you feel refreshed going into this next session, which is on the microbiome virome. And I'm pleased to introduce our first speaker, Dr. Ian Lipken from Columbia University. Dr. Lipken heads up the NIH-funded CRC at Columbia. Welcome.
Thank you. Is this now mic'd? Great, thank you. I'm going to be representing the work done in our center over the past year and a half. It's hard to believe it's been that long. I'll be trying to cover the project with respect to the bacteriome and the virome, which are largely now done in 3 compartments, as you'll see. Metabolomics with respect to methods was covered by Oliver yesterday. I'll talk a little bit about some of the work that we did previously and what we've now done and how those 2 sets of results compare. A new project in epigenetics that is a little fanciful, but I think is very interesting. And I'll close with talking about serology, which I think will be essential if we're going to fully explore the issue of the microbiome, the virome, and the fungome in these disorders. I'm representing a large number of people here. The basic scientists are in red, and in gold we have the clinicians. There's no value judgment there. And then the blue team, right, which is primarily administrative and also to some extent scientific. And as you see, we're distributed across the United States. We've been working together, at least in part, for many years. Francis Collins this morning alluded to the early work on XMRV that we undertook. It seems like a very long time ago now, and I guess it was. But that's what you see here is the XMRV study, which began really in 2010. Then with funding from the Hutchins Family Foundation, there was an initiative known as the Chronic Fatigue Initiative, which led to the collection of a wide range of samples, as well as clinical data, an extension study that was a smaller subset, and then finally, with support from NIAID, some additional sample collection. We've been storing these samples now for several years, and this has many advantages. It allows us in this biobank to examine in cross-sectional and to some extent longitudinal analyses, blood, saliva, feces, from all of these groups going back over a period of several years. We have normal controls, and we also have some controls who are individuals with multiple sclerosis. We don't have blood represented at all of these time points, but as you'll see, time 1 to time 4, which is a span of approximately a year, we tried to subdivide that into quartiles and collected at least saliva and feces. We have 3 major projects. The first one is the microbiology of these disorders. We look at bacteria, viruses, and fungi. We've completed the bacterial and virus works in large part for direct detection. Looking at feces, saliva, and blood, we have not yet done any of the work on yeast, however. We've begun to do metabolomic and transcriptomic studies. I will not be talking about the transcriptomic studies today, though I will talk a little bit about some RNA data. And then we have the third project, which is focused on clinical aspects of translation, where we look at the impact of exercise and orthostatic challenge on the pathophysiology of ME CFS. And then we have a database study which allows us to refine criteria and look for various insights which we might find by looking at endophenotypes. The exercise work has been largely led by Dana March in our team and Susan Levine, and more recently Jose Montoya and his group at Stanford. The orthostatic work is led by Cindy Bateman. Our emphasis is on rigor and reproducibility, and I think you'll see that because we have these databases and clinical sample sets, we can really adjust the whole issue of reproducibility because, as I say, we have samples that have been collected under different initiatives. I'll start with talking about the bacterial work, largely done by Doranaj Zakal and Kinji Yu. And I will be presenting the CFI study, the original work funded by the Hutchins Family Foundation, and what I'm referring to is the CFS study, which is the larger study that is ongoing now. If we look at these sorts of metagenomic analyses, so this is not 16S, it's full genome sequencing, we have in the original CFI study 50 cases and 50 controls. In the CFS study, we've looked at 106 cases and 91 controls, and this Venn diagram illustrates what is common in both of those studies, and this is where we want to focus. So the green arrows indicate those bacteria that are present in higher concentrations in cases, and the ones that are in gray and pointing down are the ones that are lower. So there are a few leads with this fecal study. First is that C. boltii and R. navis are associated with inflammatory bowel disease. Navis is also associated with degradation of mucus that could have an impact on the ability of molecules to transit from the gut into the systemic circulation. In addition, we have a reduction in levels of butyrate producers, and butyrate producers are very important for controlling immune system function and apoptosis. If we predict pathways based on the bacteria that are present, again focusing on the overlap between the original 50-case, 50-control study and the 106-case, 91-control study, you can see that the common bacteria are associated with pyrimidine ribonucleosides and vitamin B6. If we look at those patients who fall into that category that Sue Levine was talking about earlier this morning about irritable bowel syndrome, we find that there are specific differences here. Again, looking at the overlap between the smaller study and the more recent study, 9 bacteria come up, and Clostridial species, again, the butyrate producers are reduced in ME/CfS with irritable bowel syndrome. If we look at the saliva, we're finding that there are certain bacterial species that appear to be overrepresented in ME/CfS. They are frequently associated with both oral and GI disease, and we see a reduction in Neisseria and Veillonella species that are more abundant in healthy humans and mice. So there is evidence, at least in this case control study, both of these case control studies where we can look at that. We unfortunately don't have saliva from the earlier CFI study. You'll see that there are things that are predictive of associations with a number of different diseases, suggesting that there may be, in fact, dysbiosis, at least in some of these patients. And here's more evidence of that. We're looking at salivary bacterial metabolic pathways. You see reduced arginine in ME/CfS, and in this instance, we have a prediction based on the bacterial species present that is associated with a metabolomic disturbance that was found by Oliver Fien and his team. Now, viromics, which I've heard a lot about today. Jose gave a wonderful talk, I thought, about herpes viruses and the potential important. Dan Peterson did as well. We held a lot of hope that something was going to jump out of this work. So we've now done fecal, salivary, and PBMC viromics. These are all the different viruses that we've detected. I'm not going to run through them all. I will tell you that if you use purely statistical criteria, there is no one agent that jumps out, whether it's a herpes virus or an adenovirus or any other virus. That's not to say that viruses can't be implicated. It simply means that at the time we sample, the compartments we sample, we are not finding it. And I'll be talking at the end about serology and the ways in which this can give us leads that we can't find this other way. And I know that many of you treat patients with things like valganciclovir and that these have been effective. And I will tell you that in the work we now do, we don't yet have a CLIA lab, but we will have one shortly. When we analyze PBMC and we find evidence of herpes virus infections, whatever type of herpes virus infection it is, many clinicians, based on this research-grade test, have treated these patients with anti-herpes drugs, and they have reported response. This is not a controlled trial, but I would certainly be sympathetic to anybody who had such a finding who wanted to do such. It would make sense, at least biologically. Specifically focusing on herpes viruses, HHV1 through HHV8. Again, nothing jumps out in terms of statistical significance with respect to herpes viruses in saliva, PBMC, or feces. We have not specifically looked at NK cells. that is something that I think we might wish to do in the future, but this is a progress report and gives you some sense of where we are. Now, Oliver Fien talked with you yesterday about the approach that he takes at UC Davis and the West Coast Metabolomics Center to looking at metabolomic profiles, and I'm not going to replicate, and I couldn't replicate what he talked about, but I will show you that it is important to maintain updated databases. So in this slide, you see this original CFI study, which was the first metabolomic work which we did, and more recently the CFS study, done using the same instruments, but because there have been database updates, it can be very difficult to compare the results of these studies. It requires some finesse. But if you look at the end of the day at what came out with the CFI study, what came out of the CFS study, and you focus on those 7 metabolites that were common in the Venn diagram, you can see that they relate to phospholipids and sphingomyelins. These have been reported, obviously, in previous work by Navio, Narzikal, Germain, and so on. We have some new findings that came up in the larger study that I'm going to represent here, and that is the presence of these molecules known as plasmalogens. These are reduced in the brain in Alzheimer's and Parkinson's and in MS. Whether this has a relationship to disease or not is something we need to explore. Again, these are findings that we're only just beginning to work up, and we need to understand them in better detail. We're presenting the data slightly differently here, but again, if you compare the CFI study, which is on the left panel, versus the larger CFS study, you can see again that with the larger sample set, we're finding these molecules as known as plasmalogens. And if you have more questions about them, I will defer to Oliver, wherever he is, to talk about them. Now, I was really interested in trying to approach the issue of epigenetics, and we really didn't have any support to do this. So we did a very small pilot just to see it was going to pan out. So what we've done is to look in a very, very simple way at work that we could pursue with the original CFI pilot of 50 cases and 50 controls, and very simply looking at methylation differences, with the reasoning being that if a gene is hypermethylated, it's not going to be expressed to the same levels. And you can see here the locations of the methylation sites relative to the promoters, the methylation differences, and so forth. I'm going to focus only on one that we've had a chance to try to test as a hypothesis with respect to using things like transcriptomics. And what we've done here is to develop real-time PCR assays that allow us to ask questions. And these are all really relating to disorders that are well known to be associated with a variety of intriguing disorders. And what I find interesting about them is that you can study them from a pathway vantage point. And I'll show you that now through a cartoon. So here you have iron, which is free in the circulation. It gets bound to transferrin. It gets bound to a dipheric transferrin receptor. You can see here in this inset the RNA data. So you can see not only do we have a methylation difference, but we also see reduced levels of transcripts expressed from those genes. And although those differences are modest, as you'll see, they're all linked in the same pathway, and the cumulative effects may be important. So the iron binds to the receptor. It's all internalized through a clathrin-coated vesicle. And these are the two in bolded here that are important in terms of the clathrin-coated vesicle. They are then brought into the endosome. There's another protein that's important for the release of those molecules. They are released into the endosome. They're pumped out. They go into the cytoplasm and then wind up going into the mitochondria, as you see here. So we would predict, based on what we've seen with the epigenetic analyses, that there are lesions at the level of 4 of these important factors that are associated with iron transport. And iron, of course, is very important for oxidative metabolism, but it's also important you need to control iron to prevent oxidative damage and so forth. So this is a very early pilot sort of a presentation, and I don't have any more data literally than I'm showing you. But it is interesting, and it shows what can be achieved using these sort of epigenetic approaches. When we failed to find anything that was truly extraordinary using direct methods for detection of infectious agents, which included positive selection for viruses, bacteria, and so on, we began shifting to use of serological arrays. We've recently built a chip in collaboration with Roche Biosciences that allows us to map the entire proteome of all viruses. We've done similar sorts of things with various bacteria. It's a fluorescent assay, and you can see the size of the slides here. They're really fairly straightforward to use. Last year, we reported the implementation of this for diagnosis of tick-borne diseases. The arrays demonstrate the entire proteome of the relevant genes for Borrelia species, Babesia, Anaplasma, Ehrlichia, Rickettsia, Powassan, Heartland virus, and viruses that we've begun to discover in ticks that are unknown significance, and a total of roughly 170,000 peptides are required. Using this sort of an approach, you can see here that you get a very nice sort of normal distribution as you get binding to these peptides, and that lets you define the critical epitopes for these individual agents. We can use these to not only diagnose the primary consideration of the clinician, which in most of these instances with Lyme disease, but we can also find co-infections, in this case, Babesia and Anaplasma. And it's possible to move these onto even smaller arrays. So those large arrays cost roughly $500 each to use. This is about $5, and it works very, very rapidly, and the readers are cheaper as well. This is an example of how we've been able to move these, and you can see that you get very, very clean data with this pep-per-print array. It's even cleaner than what we got with the larger slides. Now I'm going to show you how we've used this more recently to explore a disease that hasn't been explained, that's been a lot in the news, called acute flaccid myelitis. This is an illness that presents with acute flaccid limb weakness. It sort of looks like polio. Magnetic resonance imaging shows changes in the spinal cord. It should span at least 1 or 2 vertebral segments. Almost all of these children have a history of a febrile illness. Most of them have a respiratory disease. Some also have GI disease. This is the epidemiology. We tend to see peaks of this disorder every 2 years. This is not yet understood. We decided to approach this in collaboration with Steve Overstee and Fei Ng at the CDC, initially by using a method called ViraCap-seq, which is a very sensitive method for viral sequencing in spinal fluid. And with 2 AFM cases, we were able to find and recover nearly complete genomes for enterovirus 71 and enterovirus 30. But the vast majority of the cases that we studied, we were unable to find any nucleic acid. So we designed a serochip to address the same issue by looking for something like a hit-and-run phenomenon. And here we've represented findings for binding with the serochip to enterovirus A, B, C, and D types. And you can see here now that the vast majority, it's actually 87% of the children with a clinical diagnosis of acute flaccid myelitis have antibodies directed against type A and type D enteroviruses in spinal fluid. We found none in children with Kawasaki disease, which is a vasculitis of children. We found none in adults who didn't have AFM. And we found in a very, very small set, roughly 4% of children in Gorakhpur, India, who had a bacterial meningitis and had high titers of these antibodies in spinal fluid, we were able to detect antibodies to enteroviruses, and they were not to A and D enteroviruses. So based on this sort of data, we feel that we have a fairly strong case now for making enteroviruses the leading candidate for causative agent and triggering this disorder known as acute flaccid myelitis. Now, what are the implications of this sort of work for ME/CfS? If there are candidates that we or anybody else finds, you can develop these sorts of arrays and you can use these to test those hypotheses, because the agent may no longer be present, but you may nonetheless have evidence that the agent was responsible for triggering the disease. So the next phase of the microbiology, which is Project 1 in the Center for Solutions for ME/CfS, is to complete the fungal work in the feces and the saliva and the blood, and to pursue serology with these and other candidates. I don't do this work in a vacuum, so this is a list of the various people who have done this work, and I'm acknowledging the funding from a wide range of organizations, including Hutchins, who's unfortunately misspelled. Thank you very much. Are we doing questions or not?
Yeah. Thank you, Dr. Lipken. Are there any questions?
Jose Montoya.
Ian, great talk. Ian, on the virome, saliva, stool, blood, where you didn't find significant differences, have you analyzed that data by severity?
No.
Thank you for your eloquent answer.
Jose, we, the viromic data is 24 hours old. I brought it specifically for you.
That's amazing. If you don't mind, add the severity to the analysis. Thank you. Thank you.
On the right.
Thank you. Janet Flint, I'm a patient in North Carolina. We have a lot of ticks, and I'm an outdoor type person with dogs, lots of dogs. So I didn't know if you tested for ehrlichiosis, because I know one of my dogs had a sensitivity to that, and I joked with my vet that maybe I should get tested for that too. Was that in your, when you were talking about the chip that's been developed for the different tick-borne diseases or bacteria?
So we have a serological tick that simultaneously addresses Ehrlichia, Anaplasma, Babesia, European and US-based forms of Borrelia, and we hope in the not-too-distant future Stari as well, once we have a better notion as to what that actually is. We have not run these on any of the samples from the ME/CfS cohort that we have. We developed these with the intramural program here at NIH with Adriana Marquez, primarily to look at people who have known tick-borne exposures who have not responded to treatment.
Okay.
This is certainly, though, going to be part of that, as well as a number of autoantigens and herpes viruses and other things of that nature.
That's great. And is it commercially available? Can I pay for having that test or ask my insurance company?
It is not yet
advance?
commercially available. We're trying to take it through CLIA certification at New York State. We hope that the viral panels will be available through New York State within the next 6 months.
Oh, that would be great. And I noted that the government agents, I can't think of the name right now, sorry, but they've noted Babesia on the blood bank screening. Are you adding more? Are you involved in
I'm not involved with the FDA or blood banking.
Because I know I've had surgeries and I've had blood, packs, and so one of my thoughts was maybe that was one of the causes. So anyway, but if anything else you could do there would be helpful.
Thank you very much. Jeff?
Beautiful talk. Just wondered whether, this may be a short answer as well, have you had a chance to look at CSF in terms of the virome or bacteriome, and if so, what sort of numbers?
So this is a huge problem. So Oliver ran spinal fluid and did a metabolomic study. We didn't have sufficient controls for the reviewers, so we've not published even the metabolomic part. We would love to do that, and actually we've done a lot of spinal fluids now all over the world, and It's remarkable. The sensitivity of VirCap-seq and BACCAP-seq is equivalent to real-time PCR because you're covering the entire virome, the entire bacteriome, and it's positive selection. So if you want to do some of that, I was talking with Avi about that earlier, we would love to do that with you.
Yeah, we have a collection as well of CSF.
Perfect, thanks.
Yeah.
Yeah.
On-stage collaborations. All right, thank you, Dr. Lipken.
Thank you. Yeah.